Hi, in this video I am going to talk about industrial fermentation media and sterilization. Industrial fermentation is done in different steps. It begins with the isolation or identification of microorganisms with the potentially interesting industrial characteristics. This microorganism can be either isolated from the nature or collected from culture collections. And this microorganism is then modified genetically to improve its productivity and other characteristics. And after this strain improvement, what we are going to do is to culture this microorganism or grow this microorganism in the specific media to get the products. So in this video, I am going to talk about the media which we are used in industry and how to sterilize this media. So fermentation media is essentially the culture media. The basic function is to satisfy nutritional and energy needs of the microorganism. The nutritional needs are the requirements for the carbon, nitrogen, and basically the micronutrients and macronutrients should be there in the media. Sometimes the microorganism needs special nutrients for its growth as well as for the product formation. And there is a need for energy, and in the most of the cases, the microorganism get energy from the carbon source, and the exceptions are there, like phototrophs, they are getting energy from light. So the basic function of a fermentation media is to satisfy the nutritional and energy needs of the microbes. Along with, it should provide substrate for the product synthesis. So in industry, we are looking for the product. So the substrate for the product should be there in the media. And how the fermentation media is different in laboratory and industry. When we think about the fermentation media in industry, the thing is that we are producing it in a huge scale. So the fermentation media in industry should be cheaper and it should be available in huge quantities and it should support the maximum product formation. So in this video, we are going to discuss only about the fermentation media that we are going to use in industry. And its characteristics are pretty much different than the media what we are using in laboratory. So fermentation media, its basic function is to provide adequate nutrients and energy for the microorganism. The adequate nutrients means it should support the growth of microorganism and should support the product formation, and it should be industrially useful. An industrially useful media should be economical, should be easily available, and there are several criteria to define a media which is industrially useful. And another characteristics what we are looking is like it should be capable to bring out full industrial potential of the microorganism. Industrial potential in the sense the productivity. The media what we are using in industry should bring out maximum product from the microorganism. The maximum productivity should be there so that the if the product is high, the fermentation process will become more economical. And the importance of fermentation media in industry is like it is one of the major cost determining factor, especially in case of the low value, high volume products. The major cost determining factor is the fermentation media. But in case of high value, low volume products, fermentation media is not contributing much to the cost of the product. And sometimes the cost for the fermentation media can go up to 60 to 80% of process expenditure. The whole process expenditure up to 60 to 80% is contributed only by the cost for the media. And I think you are very much familiar with the term up to. So in some cases it may be 5%, some cases it may be 10%, but in rarely it can go to 60 and 80% of production costs. And the fermentation media is different based on the scale of production. There are different scales of production. There are laboratory scale, pilot scale, and industrial scale. In this figure, you can see this is a small fermenter. This is something the laboratory's fermenter look like, and this is a pilot scale, and this is the industrial scale. The laboratory scale production usually have an volume of like say 1 to 10 liters capacity, and so it's a low volume production. In the pilot scale, the volume may go from 25 liters to 100 liter, And in industrial scale, the volume of fermenter will be from 100 liter to 1000, 10,000, and some case say lakh liter, something bigger will be in industrial scale. So when we are using a medium in industry, if it is a laboratory scale or the small scale, sometimes the biopharmaceuticals are made in small scale fermentation in industry. The small scale media characteristics are different from the large scale. So if you are making only for 1 liter of medium, you can actually make it using purified chemicals or expensive chemicals because it's only a matter of 1 liter. But if you are making a media for 10,000 liter capacity fermenter, you cannot afford that because the raw materials may be needed in tons. In 1 liter fermenter, you need only in maybe in milligrams or gram. But if in a 10,000 liter fermenter, this need required in huge quantity and it will be more expensive. So depending on the scale of production, the characteristics and type of media will change, and the media will vary based on the end product, whether we are producing a biomass or a primary metabolites or secondary metabolites, and each of these specific products we are using different media. For example, Saccharomyces cerevisiae can be used for single cell protein production and alcohol production. Both are the same organism. We can use for biomass or for the primary metabolites. But the media we are using for the single cell protein production from Saccharomyces cerevisiae is totally different from the media we are using for the production of alcohol. Even we are using the same organism, the media, depending on the end product, the media characteristics will change. So media characteristics will change depending on the scale of production and the end product. And further, in one single fermentation, depending on the stage of fermentation, the media characteristics may change. In the most of the fermenters, in industrial fermenters, there are seed fermenters. Seed fermenters are the fermenters which is used to produce inoculum for the production fermenter. So there is no product formation occurs in seed fermenters, but the growth of microorganism occurs in the seed fermenter. And in some cases, they have pre-seed fermenters, which is actually used as a seed for the seed fermenter. And once the microorganism grown in the seed fermenter, then transferred into the production fermenter. So the basic function of the seed fermenter is to expand the inoculum. In the laboratory, when we inoculate something, we will use a wire loop or something like that, and we are inoculating in very small volumes. But in industry, the size of inoculum is huge. Sometimes it goes like 10 liters or maybe 1000 liters. The inoculum should be that much big. So seed fermenters are used to create such type of inoculum. And the production fermenter, the use of the production fermenter is to produce the product. So the basic aim of seed fermenter is to expand the inoculum. Production fermenter is used to get the products. So the media used in seed fermenter and the production fermenter is totally different. The components in seed fermenter support the growth of microorganism, but the components in the production fermenter medium support the product formation. So within a fermentation, depending on the stage of fermentation, the type of media used may change. And these are some facts about the industrial media. One thing is that in industry, we use liquid media generally rather than solid media. There are several reasons for that. One thing is that when compared to the solid media, liquid media require less space. We don't need much space because if you are using the solid media, there should be provision for aeration. So if you are fill a huge container with the solid media, the microorganism may not grow. It may grow only in the surface because of the lack of aeration. So to ensure aeration in solid media, it requires huge space. But liquid media is not like that. To aeration is easy, and for liquid media, it's amenable to the engineering process. Like if you want to change the pH of the media, you can add the acid or alkali and give it a mix. But liquid media, giving a mix is very difficult when compared to the liquid media. Or then sometimes we need to transport the media from one container to another. Maybe the sterile media we have, we need to transport it to one like autoclave to another place. Liquid media we can actually pump using pipes and a motor. We can pump it from one location to another. But solid media may need a truck or something like to transport. So it is easy to use the liquid media. And to solidify the media, most of the cases we need some solidifying agents like agar. Adding this solidifying agent to the medium is an additional cost, so we don't prefer that. So these are the reasons why we are not using solid media so commonly. I'm not telling that we only use liquid media in industry. I'm telling that liquid media is generally used. Solid media is used, solid state fermentation is there, which is used for the production of amylase and production of mushrooms. Solid media is used, but there are some difficulties when we are using the solid media. So using liquid media is easy, so most people on most fermentation use liquid media. Another fact is that most of the industrial fermentation media is made up of unpurified raw materials. In laboratory, when we making a nutrient agar, we will weigh the peptone, which is a purified chemical, yeast extract or beef extract, which is also pure and comparatively sterile. Use NaCl. If you look at the bottle, it will say 99.9% pure. We'll use distilled water. So everything is pure chemicals in the laboratory. But in case of industry, most probably we are using something derived from plants or animals, like byproducts or the waste products of other industries. The reason is that industrial fermentation need to be economical. In order to the product need to be cheap for a market, the pricing is important. So if the product is cheaper, people will buy more. So it will be more economical. So we have a economical factor in between in it. So the industrial media should be cheaper. So we are basically using byproducts or waste products from other industries. If I say some examples, sulfate waste liquor is a waste product of paper industry. And some of the fermentation is using this sulfate waste liquor as a raw material. So actually we are using this waste management along with the production of value added products. Another example is whey. Whey is a waste product from cheese industry, which has so many environmental problem if we are releasing it into the nature or the environment. But some fermentation use this whey as a raw material. So it's a cheaper product. You don't have to, most of the time, you don't have to pay anything for the whey because it's a waste product. Sometimes the industry will pay you for taking that waste away. And the media will be formulated using different components. If we are taking one waste product, we may be using some pure chemicals to enrich the media or formulate the media. The reason why we are using the pure chemical is purely because of the expense. If you are trying to use the waste, this pure chemicals for the industrial fermentation, it will be very expensive. So the requirements of an industrial media are it must satisfy the needs of organism in terms of carbon, nitrogen, minerals, growth factors and water. So it should have basically everything for the growth and product formation by the organism. And it should be in correct proportion. And the proportion is important because if the microorganism should not get the all components in correct proportion, it may not grow, or it has a standard growth, or it will grow, and after the growth some of the media components will be left as unutilized, and it will end up in the waste, and you need to pay, the industry need to pay for the waste management or the wastewater treatment. So it should be in correct proportion so that the all the components will be used and waste generation will be less, and it must not contain any materials which are inhibitory to growth or product formation. So these are the requirements of an industrial media. So we have discussed something about the industrial media, what are the main requirements and the general features of the industrial media. Thank you so much for listening.